Hi, I'm Katie. Thank you all for coming to my talk. So we all know that invasive species can cause significant trauma to native ecosystems, whether by outcompeting native organisms, acting as novel predators, or even just by being problematic novel prey. Invasive species have wreaked havoc all over the world. An important part of understanding how invasive species alter ecosystems is uncovering any immunological effects they might have on native species. Many invasives harbor novel pathogens which immunologically compromise native species. What I wanted to know was, can invasive species cause more subtle, sublethal immunological changes in native species? In order to explore this idea, I tested the immunological effects of an invasive species on a native animal, the eastern fence lizard Sceloporus undulatus. In its southern range, the eastern fence lizard coexists with the invasive fire ant Solenopsis invicta. The gray shaded area on the map here is the range of the eastern fence lizard, and the colorful areas in the south are where fire ants have invaded. Fence lizards and fire ants prefer the same disturbed habitats and encounter one another frequently. Here's a fire ant mound right next to a lizard, actually. And fire ants attack and kill lizards, acting as a novel predator with as few as 12 ants able to kill a lizard by prying up their scales and stinging beneath them. But in areas with fire ants, we know the lizards eat a lot of the fire ants, even though they can sting and envenomate even while being eaten. So it's an interesting system where the fire ants are both a novel predator and a novel prey item. So how have invasive ants affected these native lizards? There are the direct effects. Lizards in areas with fire ants have increased mortality and their populations recruit fewer new members. This is most commonly associated with predation by ants, but frequent sublethal encounters with fire ants can also likely contribute. This may be mediated in part through their altered immune response. Lizards in areas with fire ants also have higher baseline and stress-induced glucocorticoids than lizards in areas without fire ants. This could be due to population differences, but we also know that direct fire ant encounters elevate CORT, making it likely that the elevated CORT may be attributed to frequent fire ant encounters. We also know from a previous study that lizards living with fire ants in the wild have significant immune differences compared to lizards in areas without fire ants. The immune differences in lizards in areas invaded by fire ants in the wild appear to be tailored towards the specific stressor, with elevations in measures such as anti-fire ant IgM antibodies and basophils, both of which might be useful in dealing with envenomation. However, the study only showed an association. We still don't know if something else could potentially be causing the changes. For example, complement and cell-mediated immunity are commonly affected by glucocorticoid stress, so that could be playing into this. So my questions for this study were, first, how does fire ant exposure affect fence lizard immune function? I wanted to figure out if the immune differences seen in the field are due to direct fire ant exposure. Second, do immune effects vary by route of exposure? Since fence lizards are exposed to fire ants and their venom through both being stung and eating the ants, I wonder if maybe there's a benefit to eating the ants. Maybe the different route alters the immune reaction and helping them to develop some sort of tolerance or immunity to the venom. For this study, we took 52 lizards from an uninvaded site in Kentucky because I wanted to use lizards naive to fire ants to avoid the confounding effects of previous fire ant exposure. The lizards were then split into 3 experimental groups: fed, stung, and control. The fed group was given 10 deceased fire ants 3 times a week for 3 weeks, with the fire ants glued to a live cricket with a dab of honey to get the lizards to eat them. I needed the fire ants to be dead so they couldn't sting the lizards and get an overlapping exposure with the stung group, but the fence lizards will not eat deceased prey. But they will pretty easily eat a live cricket with a weird sticky ball of dead ants on it. The stung group was then stung by 8 to 10 fire ants for 30 seconds 3 times a week for 3 weeks, with the lizards' mouths taped shut in order to avoid fire ant consumption. We know from previous studies this level of exposure will stress the lizards out, but will not cause them any lasting harm. And the control group was given handling and feeding controls for both the fed and the stung group, and the fed and the stung groups also received controls for the other group. After 3 weeks of exposures, we tested a variety of immune measures, and I'm only going to go over some of them here. I looked at how much IgM antibody each lizard had against fire ants. This antibody should be helpful in responding to fire ant envenomation by binding up and neutralizing venom in the bloodstream. And we looked at white blood cell percentages overall, just by looking at blood smears, and I'm going to focus here on basophils. We also looked at complement activity by measuring titers of lytic response to a novel antigen. And then to assess lymphocytic cell-mediated immunity, I used the phytohemagglutinin or PHA skin test, which involves measuring the swelling response to an injection of PHA into the rear foot, with larger swellings indicative of a stronger immune response. And this is a lizard getting his foot measured right here. All of these graphs will show the immune function on the y-axis and experimental groups on the x, with letters above indicating different levels of significance. So first off, we did see differences in the immune responses of lizards fed fire ants versus stung by fire ants. The anti-fire ant IgM antibodies were increased in lizards fed fire ants relative to stung lizards, and I theorized that the stung lizards may have used up a lot of their IgM binding to venom from stings, while lizards fed fire ants maybe had enough exposure to trigger the production of additional antibodies, but weren't damaged enough to require them to actually use any of those antibodies. And then comparing this to the field results, in the field we actually see an increase in anti-fire antibodies in lizards from invaded sites. This may be due to the effects of eating fire ants predominating over stinging, the effect of prolonged antigenic stimulation from repeated fire ant encounters, or natural selection for lizards which produce more of these antibodies. Moving on to the basophils, we see a similar pattern to that of the anti-fire ant IgM, where fed lizards are increased relative to stung lizards. And again in the field, we saw higher basophils in invaded lizards. This immune measure responded to fire ants in the field and lab almost exactly the same as the anti-fire ant IgM. Then complement statistically in the lab appears to mimic the results we see in the IgM and basophils, with fed immunity being increased relative to stung lizards. But in the field, we saw a different pattern with lizards from invaded sites having lower complement than those from uninvaded sites. It's possible this is an effect of chronic stress or may support the idea that complement is being used up in lizards stung by fire ants, and this may be the predominant effect seen in the field. This brings us to cell-mediated immunity, which showed a different pattern than any of the other immune measures. Stung lizards showed a strong increase in cell-mediated immune response, which may indicate that the body revved up lymphocytes to deal with envenomation, or simply could be a response to skin wounding since the stings damage the skin, and this is a test of skin immunity as well. This is also the opposite of what we find in the field, and it is the only immune measure tested where the field results don't match up with one of the laboratory fire ant exposures. Lizards from fire invaded sites generally have reduced cell-mediated immune response, which we theorize is due to chronic stress, which is supported by data from other studies we have showing lizards treated with glucocorticoids have decreased cell-mediated immunity. Bringing all of this back to our field results with the tentative prediction that if immune differences seen in the field were due to fire ant exposure, we should see a similar pattern here, although it may vary by exposure. So lizards fed fire ants match lizards in the invaded area in two measures, basophils and IgM, but not in others. But feeding fire ants generally showed an enhancing effect with no immune measures decreased. Stung lizards matched lizards from invaded areas in terms of complement, but not any of the other three immune measures which had differed in the field. Stung lizards overall showed mostly decreases in immune measures with only cell-mediated immunity increasing, a response that is not seen in the field. But for three of the four measures which were different in the field, one of the fire ant exposures produced a similar change. So this supports the theory that at least some of the immune differences seen in invaded lizards in the field are due to fire ant exposure. So one of the most interesting results from the study was finding that consuming fire ants may help lizards deal with envenomation. Consumption of fire ants was shown to enhance multiple immune functions, all of which were decreased by stinging. And whereas being stung by fire ants produced a mixed immune response with several reduced and one increased. I theorize that the reduced immunosuppression effects are most likely due to the lizards using up those immunological resources dealing with venom from the fire ant stings. So it would be really interesting to test if prior feeding with fire ants actually altered outcomes of later envenomation. So thank you all for listening. If anyone has any questions or suggestions, I'd love to hear from you. Thank you.